everyone, Ashley and Owen from Team YSI, and today we have a really special guest that we're interviewing, George Church. He's coming to us from Harvard Medical School. And if you could you just please give us a brief introduction of your work and of yourself as well?
Yeah, so I'm a professor at Harvard at MIT, and we study all kinds of strange phenomena and invent technologies for use in mostly having to do with DNA and medicine.
Awesome. That's really, really cool. I'm sure that a lot of kids especially would know you for being the guy who's going to bring back the woolly mammoth. That must be really exciting. So how did that come to be?
Yeah, so some of the methods that we made for reading ancient DNA, we found that that we could get really high quality insight into the DNA of long extinct species. So that's reading it, and then it became more and more evident that to really understand it, we needed to write it, to engineer the DNA that's in the computer from ancient times into real living elephant cells. So we got some Asian elephant cells. Asian elephants are an endangered species, so you want to be very careful not to hurt them. But you can get cells from them, and then we used a new technology called Crispr to engineer them. Crispr is a way of basically designing in a computer what you'd like to have working in a living any living system. And so then we made a list from the mammoth DNA of all the things that could help a modern elephant survive in the cold, because that's the main difference. The Asian elephant and the mammoth are very, very close. They could probably have babies together that would be healthy. Who knows exactly what they would look like, but they would anyway. So the idea was to go down the list and ask what could make a mammoth Asian elephant hybrid live and enjoy themselves at minus 40 degrees in this huge ecosystem in Russia and in Canada and Alaska, where they're needed, where they only recently, 10,000 years ago, went extinct, probably due to human involvement as well as some other changes. Anyway, so the sort of things that became evident would be useful for cold would be the most obvious thing is the hair. So they had like woolly hair and long hair. And they had subcutaneous fat that was 10 centimeters thick, just really to help insulate them. And they had a special kind of blood protein, hemoglobin, that unlike our blood protein, exchanges oxygen very readily at low temperatures, while ours is more limited. And then they have smaller ears than most elephants do, so that they don't lose heat through their ears, while the African and Asian elephant, they want to lose heat through their ears, while the mammoth does not. And a few other things like that would be very helpful. So we've made 15 changes already to the genome of the Asian elephant cells.
Implanting, I guess you would say, what you have in a computer and bringing that into a living creature. How does that go in? Is it?
We've gone through a whole series of technologies over my scientific career on how to move things, an idea, to the computer, and then from the computer you design something that you build. So sometimes we'll use the computer to design and build a conventional instrument like a robot or a microscope or something, and other times we'll make machines that are so small you can't see them. Sometimes we'll call them nanomachines or molecular devices, and sometimes those molecular devices, the most sophisticated ones, are not designed; they're found in nature and then sort of adapted. And one of the most amazing ones we found in nature, most of these we find in microorganisms, where the whole organism you can't see, and inside of them are machines that do amazing things. And one of these is called CRISPR, for reasons that are just not even worth going into. It's a cool name at this point. And what it means now is more important than what it used to mean, which is it means now is that you can edit DNA the same way you might edit with your word processor a document that you're writing, or you might edit with your pencil and paper a drawing that you're making. But you can do it very precisely. And the way so this was something we found in microorganisms where its natural function is to fight viruses in them. So their viruses infect microorganisms. It seems like, why would they pick on anything so small? But the microorganisms fight back with this thing called CRISPR. We then adapted it basically cuts DNA very precisely. It cuts the viral DNA. We adapted it so that it would cut, and then we could edit at that cut to make whatever we wanted. And this is all designed in the computer. The computer decides where to cut and where to paste in, like a bandage, to fix the cut with exactly what you want. So you can bring back DNA that has been long dead and synthesize it with chemicals and then put it into these elephant cells.
Fantastic. You probably get this question often, but what's your take? I'm sure you've seen Jurassic Park, and what they do is kind of in the same realm as what you are doing. So how do you feel about those when you watch those movies?
Yeah, so we have a lot of relationship with Jurassic Park. It turns out that the DNA sequence they included in the original, that Michael Crichton included in the original book, was from work that I did during as a student. I actually, it was not from a dinosaur. It was from a bacterial piece of DNA, and he changed it a little bit, but it was basically bacterial DNA. And then the second connection is a number of our projects in our lab will make things that we're worried about escaping, and so we often quote, "Life finds a way," this apocryphal line from the Jurassic Park. But yeah, their relationships. The main difference is the DNA from dinosaurs in mosquitoes in amber doesn't seem like a very feasible way of getting DNA. But you can get it from a variety of other sources. You can get it from frozen mammoths. The record now is a frozen horse that's 700,000 years ago it died. But you can also get it from museum specimens. Don't try this on your next museum visit, but in principle, just a tiny little piece of a skin or feather or something is enough to decode that ancient animal.
So I'm really interested in what CRISPR-Cas9 can do for like genetic diseases. That's something that personally I have a genetic disease, and I'm just really curious to hear what you think is possible.
CRISPR as a technology is only 3 years old, but it's already proven that it's extraordinarily versatile. It can be used for a variety of things, including agricultural. It can work on agricultural species, plants, and animals. And in humans, it can be used for affecting gene therapy for people who have rare or common genetic diseases, as well as infectious diseases. You can treat as a genetic disease in the sense that all of us, or almost all of us, are sensitive to HIV, and so you can engineer in a gene therapy in a clinical setting resistance to that disease. For example, there are about 2,000 gene therapies in clinical trials now, and some of them will, for example, cure kids that are born with blindness, and you can inject the gene therapy into their retina, and then they're cured. And so you want to do that very early in life. Otherwise, they're cured of the blindness, but they can't. Their brain was already too developed to be able to recognize faces. But if you do it early, then they can get the full range of recovery of vision and recognition of faces. But you can also use it for injecting it into the muscle. You can cure muscular dystrophy. These are something we reported recently in published. You can also take out the blood cells, take some of the blood cells out, engineer them, put them back in, for example, to cure AIDS. You can deliver via the blood to the liver to fight hepatitis virus or to cure genetic diseases that affect the liver, and so on. The list goes on.
I like that. So, today, what kind of things do you think K through 12 educators can do to prepare the kids of today for what kind of science they'll be doing when they get into science?
The first thing is you have to be flexible because science is changing faster than ever before. So some things that people predicted would take a century to arrived in 3 or 4 or 5 years. So that's number one. It's not so much about facts as knowing where to find the facts and how to think about them, how to do science. So teaching kids, about not trusting things until you verified them. So checking facts, doing control experiments where you have a experiment and a control, where you've changed maybe one thing, and carefully measuring them. This is all basic science, but I think an emphasis on the basic principles that are true for all science, and for that matter, human investigative journalism, and so forth, is better than emphasizing facts. So there are a few facts you need to know, but you don't need to know everything because there's a vast number that are well organized now in computers. And then K through 12, encouraging a sense of wonder that not everything is known. In fact, very little is known, and so everything can be questioned, and regular folks can make, even kids can make a big contribution. We just had one of the high school students in my laboratory just won the Intel award just a few days ago, and he made a genuine contribution to the CRISPR field that we were just talking about. So a lot of these things are increasingly accessible to high school students and younger students. A lot of these things are very inexpensive now, and so getting hands-on experience is another thing that K through 12 teachers can do. So don't make it something that sounds like it's off in the ivory tower and everything's known and there's nothing you can contribute. It's more like, gee, there's a lot that's unknown. There's citizen science is a growing field where you can, determine things about your body, about your backyard, your universe, and make a contribution.
That's really great. I love that. Awesome.
So along those lines, and following your passion into space, what kind of applications does your work have on astronauts and maybe traveling to Mars?
Some critique of gene therapy is that it is inapplicable to healthy people. We shouldn't change average human beings. But on the other hand, if we go into space, all of us are unhealthy. Essentially, we all have a very serious genetic problems in the sense that we are very sensitive to radiation, which is genetically very variable. You can get almost a million-fold range of radiation sensitivity in naturally living organisms. We're also very sensitive to gravity, which is a biological and hence genetic phenomenon, in that even both the zero gravity that we have in the space station and the reduced gravity that we have on Mars. So Mars is about 38% the gravity on Earth. It's probably too low to maintain our bones the way our bodies are currently set up with a feedback system that's based on Earth gravity. There are behavioral and neuropsychiatric components that where new environments we're very ill adapted for them. There's issues of the microorganisms we bring with us. Which ones are we going to bring, and are they adapted? And other other organisms are going to bring a whole Noah's Ark full of whales and sequoia trees, and or are we going to be selective? And so there's a whole lot of different issues having to do with genetics and space, and most of the space science has been focused more on the mechanical engineering and sort of basic things that allow us to work in space, but not really thrive in space. So no space operation has been able to completely recycle. That is to say, where we can be self-sustaining without shipments to and from Earth. So there are a lot of issues, a lot of opportunities, really.
So people 30 years ago would think that today what you're doing is science fiction. What is your favorite science fiction world and why?
To some extent, our work is inspired by science fiction. We do a lot of things that people do think are nearly impossible at the time we started. We don't try to do things that are really, really hard that will ruin our life in failure. We don't want to fail, but on the other hand, there are some things that look hard that are actually easy, and that's what we're looking for. So some of my favorite recent science fiction book was The Martian, and the movie as well. As I read the book, before I knew there was going to be a movie, I would actually got so excited about the way that this guy thought about I'm not going to ruin it for anybody, but the way that the Martian, the human being on Mars, thought about his predicament, that I would check his calculations. I just really got into it, and it was really quite accurate. But the main thing I liked about it is, unlike some science fiction where the scientist is either very foolish or evil or something like that. This one really, he seemed like a regular guy, quite bright, but he saved the day. He saved his own skin in that case. And there's another one. It's not quite science fiction, but which is called Contagion. It was a movie where again a really horrible disease breaks out. Not due to scientists, due to bad practices in the restaurant industry, but the scientists slowly and surely figure out what caused it and what the cure for it is, and get rid of it. So those are some of the ones that inspire me. But I think we're constantly infused by. We mentioned Jurassic Park. There are many even negative dystopic visions that can help us avoid something that's hazardous. And in fact, a lot of the technology we develop is aimed at safety. So it's just like you have safe engineering safety for automobiles and for bridges and buildings so they don't fall down in the high winds. You need similar safety for some of the biological scenarios. And the science fiction writers sometimes are better at thinking of these negative scenarios before the rest of us do. And I think that's quite inspiring.
So, if you were designing a World's Fair today, what kind of things would you envision would be in it?
I think it's harder today than it was then because we're just surrounded by so many miraculous technologies, and most of the ones that you can see have been made. I could be wrong. I mean, it could be something tomorrow. I mean, we're kind of used to being able to fly to the moon, and we have kind of ubiquitous computers everywhere. We have amazing access to foods. So most of the things that I would like to represent at this fair would be microscopic, things you can't see. So you'd have to provide a way of seeing these amazing machines. Some of these machines that are molecular are more complicated than an automobile, but they fit into a cell that you can't see. You can fit billions of these machines that are more complicated than a car. So it's there was a movie once called Fantastic Voyage where you had a very tiny submarine basically going through the body. That's what these machines are like. We build things we sometimes call nanorobots that aren't quite submarines, but they do some amazing things at the atomic level.
So we just concluded our wonderful interview with George Church. Feel free to check out his websites.
And don't forget to subscribe for more cool videos.
Perfect. We'll see you again next time. Thank you.
Bye.